Hello everyone, welcome back to RPIT Academics. I am AVIKA, working as an assistant professor in RPIT Technical and Medical Campus, Bastada, Karnal. Today we are going to discuss about the slide fixation and staining. When we prepare peripheral blood smear, then after preparing the smear or film, we have to fix the slide or we have to stain that slide for the examination purpose. So, first of all, if we talk about the fixing of the films, to preserve the morphology of the cells, these prepared films are to be fixed as soon as possible after they have dried. It is very important part to prevent the slide or prevent the smear from contact with water before the fixation is complete. In this, methyl alcohol, that is methanol, or ethyl alcohol, that is absolute alcohol, these can be used. Methylene spirit, that is 95% ethanol, must not be used because it consists of water. We have to prevent this from water, so we cannot use methylated spirit. Now, if we talk about the next part of fixing the film, to fix these films, we have to place them in the covered staining jar or tray, which consists of the alcohol for 2 to 3 minutes. Sometimes in humid climate, it might be necessary to replace the methanol 2 to 3 times per day. The old portions which are replaced, they can be used for the storing or cleaning the slides. Now, if we talk about the staining of the film, Romanowski stain is used universally employed stain for the blood film that are generally very satisfactory. There are a number of different combinations of these dyes which vary in their staining characteristics. Different combinations are Gymna stain, May Grunwald Gymna. It is good method for routine work. It is thought to produce more delicate staining characteristics. Now, right stain is a simpler method, and Leishman stain is also a simple method which is especially suitable when a stained blood film is required urgently or the routine stain is not available, that is, for example, at night. Now, if we talk about the field stain, it is rapid stain which is used primarily on the thin films for malarial parasites or filaria. What is the principle for the Romanowski stain? It is a basic dye, that is methylene blue, or its oxidation products such as azure B, which is also known as eosin, which binds to the anionic side and gives a blue-gray color to nucleic acid, that is DNA or RNA. Nucleoproteins, granules of basophils, and weakly to granules of neutrophils. An anionic or acidic dye such as eosin Y or eosin B, which binds to cationic sites on proteins and give an orange-red color to hemoglobin and eosinophil granules. You can see neutrophilic granulocytes. This type of granulocytes are neutrophilic, and you can see eosinic granulocyte and basophilic granulocytes also. Now, what is the principle? Principle of Leishman stain is a polychromatic stain. It is a polychromatic stain, and methanol, what does methanol do? It fixes cells to the slide. Methylene blue stains RNA and DNA to blue or grey color. Eosin stain hemoglobin, eosin granules, they are orange red in color. pH value of phosphate buffer is very important in this. Now, if you talk about the staining procedure, how does we stain the slides? Thin smear are air dried. Firstly, we air dry the thin smear. Then, we flood the smear with stain. We add huge amount of stain over the slide. Stain for 1 to 5 minutes. Experience will indicate the optimum time. Add an equal amount of buffer solution and mix the stain by blowing an eddy of the fluid. Now we will add buffer solution. Leave the mixture on the slide for 10 to 15 minutes. Wash off by running water directly to the center of slide to prevent a residue of precipitated stain. Stand slide on end and let dry in air. Now, how do we examine the blood smear? When it completely dries, examine the smear with microscope. We will take low power microscope, that is 10x scan, 10 times eyepiece we will take. Determine the overall staining quality of the blood smear. First of all, stain should not be too dark or too pale. It should not be very dark or very light. There should be no stain precipitate present on the smear. No precipitate of the staining material should be there. RBC should be appropriate color of reddish pink. Lymphocytes have dark purple nuclei with varying shades of blue cytoplasm. Neutrophils have dark purple nuclei with reddish granular cytoplasm. Monocytes have a lighter purple nucleus with a grey-blue cytoplasm. Eosinophils have bright red-orange granules, and basophils have dark purple nuclei and granules. Thank you and have a nice day. If you like the video, please share and subscribe our channel. Thank you.